Boas tardes a todas e a todos. Quero darlles a benvida a este magnífico salón da Facultade de Medicina e Odontoloxía. Nos acompaña en esta mesa presidencial o excelentísimo señor don Xosé Antón Sánchez Bugallo, alcalde desta cidade de Santiago de Compostela. Está tamén con nós a excelentísima señora dona Laura Sánchez Piñón, conselleira de Educación e Ordenación Universitaria da Xunta de Galicia. A persona que nos vai a dar a conferencia, Premio Nobel Dr. Torsten Wiesel, que estuvo xa onte con nós na Universidade de Santiago de Compostela nunha interesantísima sesión de debate, de reflexión conxunta sobre temas de política científica e tecnolóxica, tamén unha persona con unha amplia experiencia en ese ámbito. Está tamén con nós o ilustrísimo señor José María Fraga Bermúdez, decano, seguro que ben coñecido de todos os presentes desta Facultade de Medicina e Odontoloxía, o señor don Xosé Manuel Villanueva Prieto, que é xerente do consorcio, que impulsa este precioso e ambicioso programa ConCiencia, o profesor doutor don Jorge Mira Pérez, que é comisario precisamente do programa ConCiencia, e o profesor Carlos Acuña Castroviejo, un eminente catedrático da nosa universidade, pero ademais unha persona cunha ampla, moi extensa e intensa relación co Dr. Wiesel. Vamos a proceder a un orde de intervencións que, por non alongar moito o acto, vai a ser enormemente reducido. En primeiro lugar, vai a tomar a palabra o xerente do consorcio.
Hola, bons días. A miña intervención é simplemente por función explicar por que o consorcio está aquí e por que desenvolvemos esta actividade. O consorcio, como supoño que moitos de vostedes saben, é un organismo que está constituído pola Administración do Estado, da Comunidade Autónoma e do Concello, e ten por finalidade básica a protección do patrimonio da propia cidade de Santiago e o seu desenvolvemento turístico e cultural. Ordinariamente, o concepto de cultura se asocia moito máis co que este concepto de cultura circunscrita, as actividades plásticas, estéticas, literarias, pero claro, realmente nunha cidade como a nosa, de levar adiante un proceso de dinamización cultural que non teña en conta o que precisamente a ciencia e a súa contribución á conformación da maneira de vivir que temos neste momento é unha situación absoluta. Neste sentido, o consorcio tomou o acordo de impulsar precisamente en colaboración coa Universidade do Presidente unha iniciativa como esta que confiamos en que coalles se estabilice de cara aos futuros e que nos permita, diríamos, facer como algo natural na imaxe da cidade de Santiago precisamente a imaxe da súa presencia no mundo da configuración tamén da ciencia e do coñecemento científico en xeral. Neste sentido, agradecer, diríamos, o interese que tamén a Universidade demostrou para cooperar con este proxecto e confiamos en que realmente sexa para todos vostedes de interese a actividade que neste caso se desenvolve. Nada máis e moitas gracias.
Bueno, moitas gracias. Permítame a presentación do noso ilustre conferenciante se vai a facer despois. Eu quero transmitires aquí un par de cuestións. Unha, os agradecimentos sinceros que hai que trasladar ao consorcio, a cidade de Santiago de Compostela, ao alcalde da cidade, por apoiar este proxecto e, ademais, facelo buscando, digamos, como socio e dándolle crédito, por tanto, á Universidade de Santiago de Compostela. Quixemos que este programa era, digamos, un obxectivo común do consorcio e da Universidade de Santiago de Compostela, non só servise para traer aquí a persoas eminentes no ámbito da ciencia e do desenvolvemento tecnolóxico e que pasaran por aquí, deran unha charla e máis ou menos poidéramos velos e saber que existen, senón que tuveran un contacto intenso e o máis prolongado posible coa comunidade universitaria e co conxunto da sociedade. Eu, por certo, estou vendo xente aquí, bastante xente, que non son membros da comunidade universitaria, e iso me alegra moi especialmente, porque un dos obxectivos era precisamente abrir as portas da universidade tamén a través deste ambicioso proxecto de divulgación e contacto, cercanía coa ciencia e tecnoloxía. Pero, como decía, queríamos que houbera unha gran permeabilidade cos xoves investigadores que están traballando nos mesmos ámbitos ou disciplinas de coñecemento ou afíns, que houbera debates sobre aspectos que non son meramente ou non son só aqueles ámbitos científicos especializados nos que traballan ou traballaron quenes nos visitan. E o estamos conseguindo. Isto podía ser un obxectivo máis ou menos interesante, pero se non houbera resposta das persoas que se trasladan aquí, unha resposta positiva, pois quedaría en eso, en un sono ou en unha meta inalcanzable. A outra cousa que me encanta é ver isto cheo de estudantes. Moitas veces decimos, e ás veces, desgraciadamente, é verdad, que o estudantado tan presionado por os plans de estudios absolutamente exixentes, con tantas horas de práctica, de estudo, de traballos, etc., pois parece que non lle queda moito tempo para participar en este tipo de ámbitos ou de escenarios de extensión universitaria que son fundamentais. Por eso creo que isto este cheo hasta a bandeira, se pode decir, con tantos estudantes, a min me alegra moi especialmente, e tamén este era un obxectivo de todos os que impulsamos este proxecto e fundamentalmente do Consorcio. Remato xa. O importante, sobre todo, ven agora, pero insisto, sempre é, eu creo, de educación e de respecto a agradecer a quen apoia cada unha das iniciativas que levamos adiante na comunidade universitaria, pero eu creo que en este caso hai que agradecer ademais moi especialmente pois esa claridade de ideas, esa aposta por un proxecto novo e unha aposta que ademais consume uns recursos que son importantes e que sin eles non sería posible levar isto adiante. Nada máis, estou seguro de que a todas e a todos os presentes lles gustará, sen dúbida, a conferencia que nos espera. Eu, en todo caso, pido disculpas anticipadas porque o mellor antes de que remate me teño que ausentar porque teño outro compromiso. Pasamos, por favor, adiante, nos sentamos ali e continuamos, en este caso, con o comisario do programa ConCiencia que nos vai a trasladar algúns aspectos relativos a esta iniciativa e despois a presentación do conferenciante por parte do profesor Carlos. Gracias.
Bo día a todo o mundo. Óyeseme? Penso que sí. Hola, hola. Señor reitor, moitas grazas pola súa introdución. Señor alcalde do Concello de Santiago e presidente do Consorcio de Santiago. Señora conselleira de Educación e Ordenación Universitaria. Señor decano da Facultade de Medicina. Señor xerente do Consorcio de Santiago. Donas e cabaleiros, o que ven aí é unha imaxe de marca do programa ConCiencia. É un cráneo un pouco estrano, pero revela que o programa ConCiencia pretende, ante todo, facer unha louvanza ao intelecto, en estado puro. Como moi ben apuntou o señor reitor, o abano de persoeiros de primeira fila que se quere traer aquí pretende representar todas as áreas do coñecemento. As directrices que se seguiron para a elección do cartel foron cubrir as áreas, basicamente, de todos os premios Nobel, buscar persoeiros que tiveran relación coa Universidade de Santiago de Compostela e co seu tecido investigador, e tamén buscar unha certa sincronía con feitos relevantes da ciencia española que se estean a producir neste ano. Polo tanto, a elección de Torsten Wiesel era case inexcusable, posto que ten un gran vínculo con esta universidade grazas ao profesor Carlos Acuña Castroviejo, que falará a continuación de min, e ademais porque este ano se cumplen 100 anos da concesión do Premio Nobel a don Santiago Ramón y Cajal. De feito, a charla do Dr. Wiesel vai a ir sobre a súa historia e vai nos dicir que realmente é un dos científicos non só máis importantes da historia deste país, sino que tamén da historia mundial. Para ele, o que é mellor que un herdeiro del. Torsten Wiesel declárase herdeiro do traballo de Ramón y Cajal, recibiu o Premio Nobel no ano 81, compartido con David Hubel e Roger Sperry, un traballo que ademais tivo fortes implicacións filosóficas, porque en certo xeito dinos que o cerebro plástico é que non nace completamente determinado. Eu creo que a súa conferencia vai ser bastante ilustrativa deste exemplo. Voume xa, aproveito a ocasión para comentar, xa que hai tanta xente, que o programa ConCiencia seguirá dentro de 2 semanas, o día 27 de outubro, na Facultade de Química, coa presencia do Premio Nobel de Química 1991, Richard Ernst, e rematará, polo menos nunha primeira fase, o 10 de novembro, coa presencia de Sir Michael Atiyah, medalla Fields no ano 66, a máxima distinción que concede o eido da matemática, e o Premio Nobel do ano 2004. Pois, sin máis dilación, paso a palabra ao profesor Carlos Acuña. Carlos.
Buenos días. Hay dos razones que hacen que la presentación del profesor Torsten Wiesel sea especialmente significativa en esta su segunda visita a la Universidad de Santiago de Compostela. Una, por su implicación en la formación de investigadores y apoyo a la ciencia de calidad. La otra, en su personalidad humana, científica y académica, de la que continuamos aprendiendo. El profesor Wiesel nació en Uppsala y al terminar sus estudios de medicina se trasladó en 1955 a los Estados Unidos al laboratorio de Stephen Kuffler en la Facultad de Medicina de la Universidad de Johns Hopkins. David Hubel se unió al laboratorio en 1958 y los dos decidieron explorar las propiedades de las vías visuales, lo que marcó el comienzo de una colaboración de 20 años y que a partir de 1959 continuaron en la Facultad de Medicina de Harvard. Los profesores Torsten Wiesel y David Hubel recibieron el Premio Nobel en Medicina y Fisiología en el año 1981 y con esa concesión se premiaban las aportaciones fundamentales realizadas al conocimiento de la fisiología de la visión y su contribución a la revolución intelectual en neurociencia a la que hemos venido asistiendo en los últimos años. Pero más allá del talento del profesor Wiesel en el laboratorio, su dinámica personalidad responde a la de un humanista y se completa con una clara visión de la política universitaria y en la necesidad de que la ciencia se considere como una forma de garantizar cultura, desarrollo y libertad. En efecto, su nombramiento como presidente de la Rockefeller University en 1992 hasta 1999, su nombramiento en 1998 como presidente de la International Brain Research Organization y el de secretario general de Human Frontier Science Program desde 1999, cargo en el que continúa, responden a su capacidad de asesoramiento en educación y política científica. Professor Wiesel, it's a pleasure to have you here. We feel certain that your commitment to scientific training and research support has brought you to the University of Santiago de Compostela for the second time. On behalf of the University of Santiago, I would like to thank you once again for coming. A continuación, el profesor Torsten Wiesel pronunciará la conferencia Santiago Ramón y Cajal en Modern Brain Science. Gracias.
por favor, ¿podrían apagar las luces, si sí puede ser?
Not right now. So it's a great honor for me to come here, and I'm very pleased that there are so many students here, young scientists, as well, of course, as many distinguished members of the community here. First, I'd like to thank the Rector Magnifico, who has been very kind and generous yesterday and today. And the mayor, is it, can you hear me up in the rafters, as they say? Could you raise your hand if you can hear me? Thank you. Thank you. So I just wanted to thank the Rector Magnifico and the mayor. Mayor was very nice this morning. He showed me the mayor's house, and that's very beautiful. I must say that the city, Santiago, is beautiful to visit. And I also wanted to thank no, I need. I have a light here, so it's okay. The Galician Minister of Education and Universities, who spoke earlier, and thank him for being here and for his comments. And also the manager of the consortium, the Santiago, we met earlier, and the dean, of course, of the medical school. Since we are here at the medical school, I thank you very much for organizing this. And I also like to thank my old friend, Carlos Cunha, for his introduction. And I'd like to give a special thanks to Jorge Mira, who you heard a few minutes ago, who I think is a rather unique individual in the sense that he, with a background in physics and understanding of science, is willing to spend a fair amount of time to bring science to the general audience, the public. And I think that's a very important role that he plays. And it's rather unusual to find such individuals with the energy and enthusiasm and imagination to do this kind of stuff. And I think many of you are here, I think because of because he has been so effective in his organizes television thing and newspaper interviews, and I felt, as if I was some sort of a rock star. But so it's been interesting experience. The reason why I'm here really is to celebrate the 100th anniversary, as you heard, of Santiago Ramón y Cajal, and he has a very special role in the world in which scientific world in which I live, and we in the field look upon, and this is not just because it's a 100-year anniversary and flattery, actually we look upon him as one of the important founders of modern neuroscience. And the fact is that even today he is quoted regularly, and his as you know, his elegant, beautiful, and accurate drawings of the brain is sort of become a roadmap for many of us who are trying to understand how the brain works. Just to be a little bit philosophical, maybe Spain was, I have written this speech in a way, but I often don't write speeches, so I will try not to be formal, but at the same time, since I put in the work trying to make a presentation for, I want to. So Spain was renowned in the Renaissance for its artists and great explorers of uncharted seas. And in his time, Cajal brought a spirit of exploration and imagination of an artist to the scientific study of the nervous system of the brain. Is it difficult for many of you? I was worried about the language. There are many students here, and I'm not sure. Could you raise your hand, those of you who find it difficult to understand the English? Because if it's a C on hand, because Jose has promised that if we can sort of work together to see to, because you're all here and you want to understand what, and not just be here, but actually get something out of coming here. So I want to be sure that those of you who are good in English and can understand everything, raise your hands. No hands. Would you like to have sort of an interpreter? I mean, we have about an hour, I think, and a half. So even I feel sorry of you who are standing, but it's nothing we can do about that. I think it's full upstairs as well. But would you like to have short comments in your language, or should I continue? I should continue. Okay. Okay, so I will continue then, even if the response was somewhat ambiguous. Now, to go back to Cajal, he actually wanted to be a painter when he was young. And he drew pictures constantly, even when he was supposed to be paying attention to his teacher in the classroom. So I think drawing for Cajal was his way to discover and to form a relationship with the world. He was a rather shy person initially. And if you have not read his autobiography, I don't know if it's common, but it's a beautiful, very interesting thing to read for you, particularly if you're a student, because he struggled with his family and he struggled as a scientist. And these are pictures. This is a screensaver, and these are pictures. I'm Swedish, as you perhaps know, and these are pictures from. I have a summer house on the coast of Sweden, so these are pictures from that. So you can lower the light, and you can, if you don't understand what I'm saying, at least I kind of like these pictures. So if it, could you lower the light a little bit, because I have a light here, so I don't need the light in the room. It also has the advantage that if you want to fall asleep, then nobody notices. So anyhow, so his way of, I think, and it's perhaps not so uncommon for artists, that this is their way of reaching or communicate with the rest of the world because of their nature. Now, at the same time, he was a rather boisterous and rebellious young boy. And an expression, I think, of his intelligence and curiosity, that he had this restlessness in his soul. Now, I sort of relate to that myself, because I was not a very good student and was rather difficult as a student. No, I'm not difficult. The point I made here is that to be a bit of a rebel and questioning things and so on may not be a bad attitude if you want to be a scientist and make discoveries. And certainly Cajal had that quality. Now, his father was, as you probably know, was a doctor and a surgeon. And he was very pragmatic, and he didn't like this idea that his son should be an artist at all, and more or less forced him to go to medical school, which he did. And then when he was finished, he went to Cuba for some medical position there for a couple of years, and there he got malaria, and then he got TB, and he nearly died. And when he came back to back home to Spain, he was a patient in the hospital where his father was a surgeon. And but during this time of recovery, which took nearly a year, he bought a small microscope, and which was the changes his life. Now, I'm going to show a picture of is that the right one? Probably not. Fortunately, I played around with this morning, but with patience you will get it right. So these are the. Don't be frightened. You have to look at all these pictures, but I don't know why this happened. So this is the Ramón Cajal. That's his microscope, you can see there, and that really changed his life. And he sort of describes in his biography that I mentioned before, the new world that his microscope opened to him, and the way in which his passion for observing and drawing was channeled towards studying the cellular structure of the body, its tissue, eventually focused, as we all know, on the brain. So this picture is of him at that time. Now, I think a key to the understanding of his Cajal's creativity and achievement was his ability to draw what he was seeing, to look at fixed and stained nerve cells through the lens of his microscope with a sort of unique intensity and concentration, and do it with a very fresh eye. And he used a method that actually was developed by Golgi. And as you know, Golgi shared the Nobel Prize with Cajal. And actually, I was in Pavia, where he did his work just a month or two ago to celebrate his 100th anniversary. Because Golgi was a great anatomist in classical, and he developed a method. Without this Golgi's method, Cajal could not have carried out the work he did. So they were a very important team in a way. Now it turns out, as you will hear, they didn't love each other, but they needed each other. So the conflict that between Golgi and Cajal was that Cajal, from his observation of this Golgi-stained picture, I will show you a slide of maybe the next one. Is that? Could be, yeah. This is a drawing of Cajal. So from looking at drawing these cells and looking at them very carefully on the high magnification in his microscope, he thought there were gaps between one cell and the next. And whereas Golgi had the classical idea that the nervous system was a syncytium, that is, there were direct connection between cells. And so that was a fundamental difference in the way of looking at the brain. And that was one of Cajal's great contribution, was that he realized that there were gaps, which we now call synapses, between nerve cells. So they communicate with each other not directly, but through the synapse, which the nerves ending release a chemical that then affects the next cell, which is a electrical chemical interaction. Now, so Cajal looked upon the nervous system as a dense forest, and when you look at the adult tissue, it's very complicated. So he focused very much on the development of the brain. Earlier in life, during the embryonic time, the brain is more available for study, so he studied in many different species the nervous system. And his work very rapidly became acknowledged, even if he published it initially mainly in Spanish articles. And the renowned British physiologist Charles Sherrington, those of you, Sherrington received the Nobel Prize 20 years later for his work on studying the physiology of the brain with Adrian. And he was very early realized Cajal's importance and was amazed of his way of describing the brain in his drawing as if it was alive. I mean, of course, he used dead, fixed tissue stained with this Golgi silver nitrate stain, but still he had one thing is interesting with Cajal, that he often sat in the microscope, intensely looked, and then at night at home, he often did the drawing from memory. But, you know, many people subsequently have looked at the brain using the same method over 100 years now, and the accuracy of his work is just amazing. So it's good to lie in bed sometimes and think, remember things. So I'm using this drawing of the retina to bring out one of the important things that Cajal did. That was to establish neural doctrine. It's a doctrine. Doctrines, we are always suspicious of doctrines in a way because they tend to lock us into a way of thinking. But the doctrine has been very useful for the scientists over the years. And the first principle were, as I mentioned before, that each neuron is an individual. It's like you. We are like neurons, and we talk to each other, and that communicate with each other in different ways, obviously. But the cell is the same. It communicates with other cells. So that's one point. The second is what I mentioned before, the synapse, that make it possible for cells to communicate with each other. And it's interesting that many people doubted the presence of synapse, and it wasn't until in the early 50s when the electron microscope was available that it was possible actually to prove and show that there is a gap with the synapse. But so it's kind of intuitive in some sense for Cajal that he said this is his way of thinking about how the brain works, was that it must be, cells has to be looked upon as individual. And of course, the second thing that comes of that brain is, we have billions and billions of cells, each individual, and it's how they're interconnected, the circuitry of the brain. That is really what makes us what we are. And we have, of course, a lot to learn about the circuitry in the brain, but some of the things we can address, and I will try to so as I said before, this sense that these doctrine that cells are individual and communicate with circuitry and gaps and signal travels from one cell body out in the processes. This was changed everything in the way people were thinking. Now I like to shift gear a little bit and make the point that, as I said, that Cajal lit the roadmap for the future of neuroscience. And so those of us who are interested in the function of the nerve cell and how they're interconnected, then needed this map can you see? Yeah, you can see that, right? Yeah. So this retina, you probably know that these are the photoreceptors, these are the rods, and these are the cones, the fat thing. And signals go, light is absorbed out here, and electrical signals is generated here and communicated through the synapses, electrical, first chemical and then electrical signal, to the next cell, the bipolar cell, that then comes to the ganglion cell. So people over the year have recorded the activity from the various cell types, and I'm only going to focus on what's relevant for my presentation. This is these cells called retinal ganglion cells. And so they have received information from the photoreceptor, but it's a processing in this layer here called this layer here, the outer plexusum layer. There's all kinds of horizontal cells, etc. We don't have to go into that. But there's a processing machinery here, so when you record from these cells, they don't only respond to light, they actually care about how the light is distributed in the retina. So, and my mentor was a man, then at the time he was actually a European. He fled during the Nazi time to Australia, and then he came to America. And his name is Stephen Kuffler. He was a great guy, very much a scientist, and this work was sort of carried out initially in his laboratory. And he was the first actually to record from these cells here and study how they responded to light. Now, one of the things that is important to remember is that if you stimulate these photoreceptors out here, they are connected to these cells. But if you stimulate photoreceptors out here are not connected with this cell, then if you stimulate out here, this cell don't see that signal, the light here. It only sees the light here in this area here, which is the area of reception. We call it receptive field. So a cell, and I will show you an example, a cell, if you record from a cell here, it has a limited receptive field. And the discovery that Kuffler made was that you stimulate in the very center of this area, let's say this cone here, you may excite the cells. You hear impulses, and you will hear something. It looks like knocks on, and the amazing thing was they also showed that if you stimulate outside in this region here, stimulate here, the cell is excited, and if you stimulate here, the cell stops its spontaneous firing. So I just wanted you to show you that. But for that, this is the way, now I'll come back to Mr. Golgi, and this is an impregnation of a nerve cell with a black stain, and you can see processes. The cell have dendrites and axons, and you can see them pretty well impregnated. And so this is what we call a microelectrode. This is a metal wire that is insulated except for the very tip. And you can actually record what we call the action potential. As I said, cells signal to each other through a Morse code, as if through action potential, all or non-action potentials. So, and this electrode can record. You can tap into the signals that cells send to each other. It's legal. It's not illegal tapping, but you can do that. And so if you then do that and stimulate then the eye of the animal, this was work done in the cat, and some of the work I will talk about also done in the monkey. all anesthetized and well cared for animals. And this is what I said before. If you stimulate just a cone in the center, you excite the cell. And if you stimulate the cell surrounding the center, the cell tend to stop firing. And the consequences is that here is light. If you stimulate with a big spot of light, you try to excite the cell by these receptors here, but through the connections in the retina, you inhibit them through here. So using a big spot of light is not as good as using a small spot of light. So you may perceive that. So, and I thought, just to make it more real, I will show a part of a film, which is actually an experiment that David Jubel and I did, and we just videotaped it for our record. And so this is actually not from the retina, but from the next day. But this is from a cell with this kind of properties. So now so this is the center. This is the center part that we just showed, and this is the surround, right? So first you will see the light going on here, and then there will be the dark spot here and light in the surrounding, and you will hear the cell stops. Now, you will hear when here because the animal is asleep, and when sleeping animal have this kind of firing. Okay, so let's see. The spot, you take away the inhibition and the cell fire. So this is a very good mechanism for detecting contrast. You can think about that when you now I will stop here and come back to this film in a bit, just to show you how cells respond at higher levels. So this was on the retina, and Cajal, as I said, he drew pictures from all parts of the brain and the various systems, and this is a drawing from the visual cortex, and you know he again described the different cell types and layered structure and all kinds of details we don't have to go into. But then when my colleague David Jubel and we came to Kufler's lab, and we thought it would be fun to, and here is when we came. It was about 1958, 59 when this picture was taken. We were kind of starting out our career. And this is David. I don't know, this is me. I don't know. You may not recognize. At that time, you know, in science and many times in life, you have to be an opportunist. And if there are new methods developed and way of looking at the problem, using that nobody else had tried before, then it may be a good idea to do so. And these two young guys wanted to see what happened to the cells in the brain. Do they respond like the cell in the retina to a spot of light and with a center and a surround inhibition, and so it was just kind of an exploration. We look perhaps on ourselves more like explorer of unknown territory, because nobody has been recording from single cells in a living animal using this particular method at that time in the visual cortex. So And actually, we spent 20 years together and had a lot of fun. I think science, you know, for the young of you, science to me has always been kind of an adventure. And I sort of want you to have a sense that to be a scientist is adventurous. I mean, you have to have the spirit of that in you. Now, this is just to make it kind of easier to perhaps understand. So this is just a profile of a person and looking at the screen. And the screen, I don't know if you can see it in the back, it's just squares. And each square is roughly of the size what a single cell can see, what we call the receptive field before. That is, so a given cell can only see one part, let's say here where we have a bar of light. Not if you shine light here and here, the cell can't see that. So the cells are compartmentalized. And this is true in the retina, it's true here in the lateral uniculate body. It's a relay nucleus between the eye and the primary visual cortex. So you can record from cells here, and the cell you actually listened to was one that David and I recorded from here. The receptive fields, the response properties of these cells are practically identical with those in the retina. Of course, then you ask, now what happened if you record from cells up here in the cortex? And I don't know, this light doesn't, maybe I have two. I think Carlos gave me another one. If we can know how it works. That works? Does this work? That's the trouble with my experience of pointers, that they always burn out. But anyhow, so anyhow, the question then is, how do the cells respond up here in the visual cortex? Are they kind of boring if you just have spot centers around? And so that's one of our main discoveries, was that they were kind of interesting. So what you have to remember then is that the grid that you are looking at sort of is represented up here, so that if you stimulate here, you have to record from cells in this part, etc. So this is one of the principles how the visual system is organized. This is true for all sensory systems. Tend to have this topographical organization of the brain. You have to have some order. You can't just have wire everywhere. So it's a very neatly, highly precise organization of the brain, and I will show you some examples of that, I hope. So what David Jubel and I did was then to record from this is the third one, from cells in the primary visual cortex. And I like to show you an example of that. Now, we have to go back to, I only have one copy of this, so we will go back to that cell. So we can use this, that's fine. Now, you have to bear with me now. We have to listen to this again, but it's not. So this is just the same cell again, because I don't know how to interrupt. I just wanted to, but if you're not used to listen to nerve cells, and this is the action potential you hear. Sorry. So this is the action potential you hear. You can see how the cell, the single cells, in this case, the relay nucleus respond to light stimulation, just like in the retina. And if you're a student, you may, for fun, just make a drawing of a dark light border and see how these kind of cells can enhance contrast. just a play. Another thing that I show here is that if you use a bar of light, these cells don't care about the orientation of the bar. And this is important because these are the cells that project to the cortex. And this is a cell in the cortex now. You can hear the sound is quite different. It's still an action potential, but it's just a different frequency spectrum. So now we are trying to find first the receptive field area over which the cell responds, and then the best orientation. Again, this is an experiment. This is not made up. You're sitting in a lab, the monkey is looking as you are looking at the screen, and we record from one cell in the visual cortex, okay? And we stimulate then a very small part of the total visual field in one of the rectangles that you saw on the screen before. And as you can see, this cell responds to pretty well as you move this bar vertically across. Both direction, left is a little bit better, I think. And this is my hand drawing the thing. We didn't get it quite right. The orientation isn't quite right, and the size is not quite. As you can see, it's better long. And this is now, careful now. So this is really one of the wonders, I think, in the world, that here's a cell wired up in such a way from the retina, so you can only see light bars or contours in a given orientation. And now you will see just an ordinary paintbrush moved across. You see, it's a very robust kind of phenomenon. You can question if it's worth a Nobel Prize, but you know, it still was one important part in the work that David and I did. Now, if you, now you have to go back to, I did sort of experiment this morning trying to make it interesting to you. And see if you can get the right one. Is that Cajal up in the, yeah, I think that's the right one. So as scientists, you of course like to, you write papers and you have to illustrate the findings, and this is our way of drawing this film that you saw. So this is a visual field of the animal. This is a vertical meridian or horizontal meridian. And the cell can only see this little square here. And this particular cell can only respond to 1 o'clock orientation of the bar. If you change it, you move the light and bar light, just like in the film, across the receptive field, the square here, and you get a very good response from the cell here, in this case, about 1 o'clock, and then it declines, the response. And you can draw a graph, and the peak of the graph is here, about 1 o'clock. Now, the thing that is important for you to, if you want to understand, is that within this same square, if you record from a neighboring cell, that cell often has a different orientation preference. So it may prefer an orientation that is 2 o'clock instead of 1 o'clock. And as you record, you will find that some cells prefer horizontal orientation, some 11 o'clock orientation, etc. So each part of our visual field is analyzed in terms of orientation of contours. So these cells are as if the building blocks of our visual images as we perceive them. Of course, I should have made that point before, that we are looking at the first stage we call the primary visual cortex. Cells in the primary visual cortex send cell action potential, information to higher visual centers, where then more complex, you can have cells in higher centers responding to faces and all kinds of things. And that work has been done by other individuals. But we are now looking at the first stage, how the nervous system as if code information from the eye. So this stimulated David Marr, late David Marr, who was a very talented British theoretical an early neuro-computational type scientist who also knew anatomy, was interested in the question. He said, oh, these cells are interesting. Then maybe they can be used to draw a picture of the teddy bear, like it's just one from his book. And so we said, a cell here, a cell with horizontal orientation preference will give a response, and here vertical. So he drew this picture using these kind of stuff. This of course, is a fantasy, but I like it because it sort of gives a very concrete way of looking at how perhaps single cells in the brain with a certain specific characteristic can be used by the brain at higher center to analyze pictures. So and then, of course, when we published this in the early 60s, actually the first paper was in '59. It's it's a long time ago, and it's still I still like it after all these years. Then I received a letter from a very famous British anatomist by the name of J.C. Young. Most of you have never heard of him, but he discovered the giant axon in the squid and was very instrumental thereby in the axon channels to understand the action potentials. Anyhow, J.C. Young was an interesting guy. So he wrote to me, Torsten, this is very interesting, what you have published, but it's nothing new. And then in his letter, he included this picture. Now you're supposed to laugh, but because his point was simply that Van Gogh knew already in the late 1800s that lines drawn straight is a very powerful way of picture things. So that's sort of is a I think my own view, being interested in art and having that the insight into understanding things is sometimes you can perhaps an artist maybe ahead of you, at least. So I thought that perhaps, since some of you didn't quite get because of language problems, I asked Kosh if he could make a few. Would you be willing to do that, Horst? A sort of a short commentary on. You can show the film.
No, the film is quite clear. O Dr. Wiesel pediume, antes de empezar, que, chegados a este punto, comentara en galego as ideas básicas. E a idea básica é que todo o que vemos está feito en base a estímulos individuais de diferentes células. Hai unhas responsables de ver puntos, hai outras responsables de ver orientacións. El antes amosou unha película na cal se vía como as células dun mono, pobre mono, respondían á orientación con luz horizontal e non con orientación vertical, por exemplo. Entón, ele identificou, xunto co seu compañeiro David Hubel, como opera ese mecanismo para que podamos ver. En base aos seus descubrimentos, pois un famoso médico británico, fixo un dibuxiño do oso que vimos antes. Outro colega comentoulle que iso xa estaba descoberto porque Van Gogh xa pintaba dese xeito porque el intuía que o proceso de percepción visual procedía dese estilo, non? Vendo diferentes orientacións. Entón, el pediume que concretase ben que un vai construindo a imaxe en base a estímulos individuais de diferentes partes.
You know, I think it's very important to, you know, these formal lectures are important, but more important to me is that you actually get something out of them. And so that's, I think, if there are specific things, or if you want to see the movie again, or anything of that, I would be very happy to I mean, there's it's a long story in addition that David Jubel and I, we could show that the cortex is organized in columns, so the same cells with the same orientation preference tend to have columns and so on. I'm not going to, I thought for a while going into that, but I've been so slow. It's already 1:15, and those of you who are standing up must be fed up. So I thought instead of showing you this picture of Magritte. Magritte is a very famous Belgian painter, as many of you know, and I sort of use his pictures so we can look as if into the future. I mean, it's kind of surrealist. He was, as you know, a surrealist. So it's kind of. So I have like to make some comments about what have happened since 100 years since Cajal. And now, you know, we can not only view single cells, but we can also, through optical method, we can also look at individual synapses in living animals. You know, holographic methods. There are all kinds of new optic methods that make it possible for single photon, two photon microscopy, etc. And I think that's has revolutionized. It also put a lot of pressure on the funding agencies, because in order to really do first-rate science today, cutting-edge science, you need to have access to equipment, modern tools. And because imaging is have become a very important tool for brain scientists. So because that's just one way, but if you look at fMRI and PET scans and then down to the single cell level, all the dyes now present, you can identify cells, you can actually use a calcium stain, you can actually visualize activity of cells, etc. But in order to do this kind of work, you actually need expensive equipment. So I know when my research was going on in the early days, in 60s, 70s, and 80s, my grant was moderate. The standard grant now is twice what it used to be, at least, sometimes 3 times. So the funding of science, therefore, is in a dilemma. In the one sense, you need to have and recruit new scientists and young people. But the funding of this science is expensive. And so the resources, one has to, in the government, has to start to think about things differently. And if you're really going to be competitive with the rest of the world. And as you know, the rest of the world is emerging with resources that are often surpassing what is present in the Western world. So one of the, of course, revolution in addition to optical methods has been the Genome Project, and that has had its very clear implication also in the studies of the brain. And we are still in a very early phase. I think the Human Genome Project was probably over, it was promised perhaps to yield benefits too soon. And there's no question that it is going to be and is of tremendous, it's a revolutionary importance, and will change not only basic science but medical science dramatically. And so the way how this will be carried out is still people are exploring. I saw you probably saw in the paper and in the recent article in Nature, I think it was, that came out of Paul Allen's Neuroscience Institute on the West Coast of the United States. And they have been looking at gene activation using in situ hybridization. In this way, you can, with a dye, you can actually see which genes are activated in a given tissue. So if you look at the brain, 80% of the genes, each cell in the body has the same family of genes. The liver cell has the same genes as a brain cell, but only some genes are specifically activated in a given region. So the liver genes are activated in the liver, and now 80% of the genes seem to be activated in the brain, which came as a great surprise to many of us. So, and there are no regional differences. So, people had hoped that maybe in the cerebellum would have one set of genes activated, and the forebrain another, etc. And the retina still. That turned out not to be true. So that's interesting. Now, what is the positive thing to me, very exciting, is that if you look at individual cells, you have to remember you can actually look at the genes activated in single cells. You find that in a given region, and looking at individual cells, they have different profiles. So the hope now, what's going to happen in the future, is that we will be able perhaps to have a family of cells with similar gene expressions. And that, in turn, could perhaps be a framework for understanding how cells or assemblies of cells work together in various systems. So that's I mean, some of us weren't sure that it was such a good idea to spend, because it's an enormous amount of work to get the complete map. This was done in the mouse, I should have said. And now the next proposal by this institute is to look at the human brain, and also so each of our each region of the brain would. Now we are back to Sweden, if you don't mind. So I think one thing, I'm 82 years old, but I wish I was younger, because there's so many exciting things to do in science today, and I like to encourage those of you who are in the graduate students or postdoc and early faculty that that venture. So is here, and the possibilities and the potential to make discoveries, I think, are great. So, I don't want to, I have to mention that in the interview, people always ask you about stem cells, because in the television, etc., radio, because they've become controversial, because religious groups and politicians, depending on their route, particularly in America, that has perhaps here as well, has made stem cell biology controversial. From scientific point of view, it's a beautiful system to work on, to understand First of all, how we develop from a single cell to a full human, a full body. I mean, there's a lot of questions that we don't understand. I mean, of course, a lot of that work can be done in animals, but the whole concept of having, you call them here mother cells right here, I think is and my only hope is that countries in Europe would take a position more like the one in Britain and Sweden and Canada. Is a pretty good policy letting the scientists develop their research project according to scientific criteria. And of course, I think most in the field of science, I think all of them are serious, and scientists don't want to clone humans. I mean, that's not the issue, has never been. But the potential to be able to use these cells to understand development and perhaps even evolution in time. So it's a great tool, and I hope that some resolution up in UN that should forbid doing this kind of work. In the United States, it's not as, the administration, the current administration has a limit. You have certain cell lines, but this is if you use federal money. You should know that if you have raised private money, and the state of California has raised $3 billion for doing stem cell biology. And so that can be used in spite of all the other objections. And there are private foundations have also been generous in doing so. So I think my hope is that this is sort of once the benefits of being able to use stem cell becomes obvious, I think it's like Darwin's concept of evolution has sort of won over. I mean, there are some people who are still ignorant enough to believe in intelligent design, but that seemed to me is more a ploy, some kind of political ploy. And the reason why that had a credence in the United States of America was because of, I think, that the basic education is not what it should be. So the other thing that I think many people, we are at a medical school here, and of course in medical school, and I was, as you know, I'm a medical doctor. I was trained in medical school, so I've always been interested in trying to relate the work you do to human and clinical situations. And the issues that are particularly actively researched today in a grand scale are Parkinson's disease and Alzheimer's disease, which I think real progress probably is being made, and there is no logical reason, at least it should be possible for us to develop ways of treating these conditions. Preventing them, perhaps. It's a little bit like, I look upon it like diabetes. Diabetes, the cure, insulin was discovered in 1929, or I don't forget exactly. And so people, diabetics have been treated now for 80 years or so with insulin. But we still don't understand the root cause of diabetes. It's as if the fact that you had the medicine, it didn't stimulate the scientific world to really understand the cause of the disease. But it's sort of, probably, my guess would be that we may be able to find a way of treating these conditions. Like in Parkinson, stem cells are now being produced that generate dopamine, and they are being implanted in monkeys, and the cells seem to make connections. And there's some preliminary report, actually, that this may be a profitable way of approaching it. Of course, people have tried to do implantation with human embryonic cells before, but that has never worked out. So it's still, but even if you can sort of help the patient with Parkinson's disease by this method, we still want to understand why is a selective death of these particular cells, because it's targeted just to a small group of cells in substantia nigra in a small part of the brain. It's like Huntington's disease also. And so science is, therefore you have illnesses, but behind those you have very interesting basic science problems to study. So, you know, there's a lot of effort. I mean, we like to see it more possible to do transitional research. That is, to have scientists, good scientists, basic scientists, who are working on problems related to clinical condition, and then have that knowledge transfer into specific condition. And that has never really been successful. This is true in the United States. When I was president of Rockefeller, we have a small research hospital, and I tried to find good scientists who would fall into the category of transitional research, and it's difficult. So I think we all need to think about this as a serious experimental scientific question to try to encourage talents, individuals to. So that's the other thing that I'd like to mention at the end is, as Secretary General of the Human Frontiers Program, I've been pushing very hard for interdisciplinary research. As you know, this is an international program, so you have teams of scientists from different parts of the world, from Australia and the United States, Japan, Europe, etc. Usually 2 or 4 people, can be 2 to 4. And now we have pushed very hard to have the scientists in the team be composed by biologists, physicists, chemists, etc., and mathematical computer specialists. So, and now nearly 80% of our grants fall into that category. And I have a sense that any university that is going to be successful, not only to teach students, but also as a research, identified as a good university in research, has also to become interdisciplinary. You no longer function to have chemistry and physics and biology and mathematics separate, in separate building, not talking to each other. In the future, you will have to see too that the scientists in all of natural sciences are communicating with each other. As Sidney Brenner said at one point, that all natural sciences are converging onto the life sciences, and life sciences probably will be the area of specific focus perhaps in the coming decade. Not that, you know, astronomy and physics, there are a lot of other things, obviously. I'm just saying that in terms of when you think about how we should train our students, you should see too that they are not specialized too early, and that they have an input, and also realize what the potential is if you collaborate with somebody in a different field, and that will help you to advance your science to reach. You know, we all want to be at they say, I sort of hate the expression, but it's pretty good, the cutting edge kind of stuff. So my hope is, since we have rectors and important people here, I'm talking to them as well to see too that there is a open mind to create mechanism and to make it easier for students to move between departments, for faculty who go from one department to another, you know, I was very struck at Harvard. I was involved with Harvard for 24 years and was a board of overseers since then. And they changed the name of the Department of Chemistry is now called Chemistry and Biology. And my prediction is that maybe other departments like physics also will have a similar change because of the interest of, I mean, you have to see what the young people, what are they interested in? What kind of issues do they want to study and do scientifically as well as education? And we have to modify our you know, in neuroscience, when we started the Society of Neuroscience with 130 people, and or 150. My number is 150, a member number, and now there are over 35,000 members in the United States and also from other countries. So it's and you have to as a university you have to say what's happening here. You know, we have to see too that you actually provide opportunities for young people to, since this is what they're interested in. So this is kind of a open mind attitude. I think is important for the future of science and future of any institution who wants to be at the frontiers. And I think we all want to be relevant, not only today but also as we look ahead. So I thank particularly those of you who have been standing for all this time, and for all of you who have listened to me. It had been a pleasure to come here, and I hope that you enjoyed my presentation. Thank you. Yeah, if you ask questions, I love questions.
Si alguén quere preguntar,
Feel free.
Que levante a man e acerque o micrófono. Como vedes, douvos un speech ao final, así medio filosófico, animando aos jóvenes científicos a que vos animedes a seguir na investigación. Bueno, alguén quere comentar algo, preguntar algo?
You can ask in Galician and then
Oh, yes, there is a question.
Horst will translate it for me. So. But I will. My answer will be in English.
Can you hear me?
Yes.
Yeah, okay. You have been talking about science, and you are a great scientist yourself. And my question has to do with the word scientific. I personally think that perhaps sometimes the word scientific is a too manifold word. And I would like to give you an example, and I would like you to comment on that. For example, talking about heavy metals, particularly about mercury, depending on countries, the level of what is toxic and what is not, as I said, it depends on the country. And although the World Health Organization has some standards, levels, these have been changing depending also on the politics within the World Health Organization. And for example, it is toxic 1 part per million, referring to mercury in air in Canada, but in other countries it's 10 parts per million. Does it mean that the people from Canada are weaker than the others? I don't think so, and I think that personally it's just a politic question, and I would like you to comment on scientific and the evolution and what is related to politics. Thanks.
Yeah, I think it's a very good point to make, and I think it's too often that decisions on this level of mercury or other toxic products generated by the industry is not really based upon the toxic effect, but rather, I mean, as we all know, as you measure toxicity, there is a range, a lower range and an upper range tolerable, and those ranges are often difficult to be very precise about, and if for political reason you always choose the upper range, then you get into the problems you just mentioned. Now, I fully agree with you that the scientific world is perhaps not as influential as it should be. I don't know in this country, but in the United States, the advisor to the president has always been a physicist, and I sort of, because of the nuclear issues during the Cold War, etc., my prediction is that in this century, the biologists will probably be involved, and issues like the one you raised are perhaps going to be attended more to better. Was that okay?
I have one question for you. Ramón y Cajal is very important in Spain because it is one of the two Nobel laureates we have in science. But sometimes we think, well, it is important in Spain, but is Ramón y Cajal actually important in the framework of neurosciences, in medicine, and in science in general?
You know, I tried in my open remark to say that he, and not as a flattery or because I'm in Spain right now, that he is actually looked upon as a founder of modern neuroscience. And I always, when the graduate students, he published 2 thick volumes of drawings of various parts of the brain. And when the student graduated, I always used to give them copies of those, because I think if you don't understand and know about his work and you want to be a neuroscientist, particularly interested in the brain function, then you are not really on the right wavelength. So I think it's no question. He was a difficult guy, you know, his history is kind of interesting, but I think he was remarkable. He nearly became, he was very interested in chess, and he was a period after he had malaria and TB with the microscope. He also played chess on his free time, and he spent so much time, and he was so good at chess, so he felt that if he's not careful, he will be drawn into the world of chess. So he stopped and never played chess again after he decided to go into the anatomy of the brain. And I think it's an interesting comment on him as a person. But read his book. It's if you haven't, it's kind of, he was a bad boy, gang leader, and all kinds of things. Yes. Okay, any other question you'd like to? Here is a question. Yeah.
You talk about the disadvantage of have frontiers or boundaries between life science. And here I think the problem is even heavier because you have inside every subject, let's say chemistry, you have analytical chemistry, physical chemistry, inside chemistry. Then you have boundaries between chemistry and physical and mathematics and so on. And I am sure, or I think, as you say, that the future is with no boundaries, no limits between the science. But here we have more than 100 years of well-established boundaries, and I think that government or people who has the power has no idea, or maybe we are not doing very well this division. And just as an example, we have research institutes here, and they are supposed to born with this aim, with this objective of putting common different knowledge, but we cannot even talk to each other. We are just living in different floors of the same building. So I don't know if there is any trick or advice that you can give us to try to go on this direction.
Yeah, I think you have to I mean, scientists, I mean, we shouldn't blame administrations. Over my experience is that scientists are quite conservative as well. And so when you have new ideas and want to change things, the resistance often comes from local departments or so on. So I think the future has to be that as you plan, you need to give openings for more cooperation. I know at Berkeley they had a very strict sort of segregation. They built a new building and they put scientists from different groups together, and that's probably the easiest way. And then the old thing kind of disintegrates in time because it perhaps cannot survive. I'm involved in Okinawa. There is a institute, graduate research university being built that Sidney Brenner is in charge of. And we tried there to again build it in such a way that it's natural for physicists and chemists to work with life scientists. Structurally, you have to build it in such a way that not people are in one building here and in one field and another field, another building, but try to build bridges and see to that people can, and the best communicators, I think, are the students, and as long as you give students a postdoc opportunity to go to different departments and interact, then somehow the professors realize that maybe they have to change. So there are ways in universities to create assemblies and groups of people who have a broader interest to stimulate them to create awards for people doing interdisciplinary work. But I think what's exciting is that we, in life sciences, realize that chemists are not all bad, or physicists. I mean, it has been a certain reluctance. I talked to a chemistry professor yesterday, and he said that life scientists, they don't like us. So I said, it's not so. They are just afraid of you because chemistry and physics kind of sometimes have tremendous intellectual challenges. And I mean, I know myself, if you're not good in math and something, it may you hesitate to make a fool of yourself with somebody else. But if we, let's say, we are all limited in these different ways, and let's talk to colleagues who have different talents, and maybe together we can do a better job. That's kind of my attitude. Could you speak closer to the thing? Maybe it's not on.
Well, the other day we were talking on a lesson we had about, and we were discussing about a video we saw about brain's evolution. And our question...
Pollution?
Brain evolution.
Evolution, I see.
Yeah.
Okay.
Well, and it was, for us, was incredible how a little, just a very young brain was able to create thousands, well, millions of connections just with such a speed and with such, so easily, that we were asking ourselves what we were doing between the age of a baby just born until the age of 10 years old, more or less, that capacity just was getting less able to create that kind of connections. So then, wouldn't we, now the government, and not only Spanish government, just but others, even in Europe, it's a great question about our succeed at studying. They are complaining about that we don't get results in different areas, not only in scientific world, but in many of them. And our question was that wouldn't we change the way of learning. Wouldn't we needed to learn more at the child's time? Not letting a child just playing. It's okay, but you can play and also learn, and you can take concepts easily.
Maybe, George, you could summarize the question.
I think that the question is, provided that our brain is more able to make connections when we are young, shouldn't we learn more when we are babies and children? This is the question.
Yeah. As I saw I was interviewed yesterday by a journalist. And she made the point that we are not optimally developed. That is, and there are good reasons to think that we are doing our childhood perhaps when we are particularly sensitive for developing our brain, both in our sensory system and also in motor system, that we are maybe not using all the means available. We don't have time now to go into that, but there are some good examples for that. But at the same time, you as adult, you are still also, you can learn new languages, you can recognize new faces, and so the plasticity of the brain remains with us all through life. It's just that early in life, during the first maybe few years, the brain not only can be plastic, but it also, if not stimulated, can have regressive effect. You can cause damage. And this is why this study about cataract, if a child is born with a cataract, even if the nervous system is ready to be used, and not having normal visual stimulation, lead to that connection from that eye is lost, more or less, in the visual cortex. So the cells that you heard before only respond to the eye with no cataracts. And so this is a period, so nowadays children are operated on soon after birth in order to have exposure to normal light. And so this is a kind of, we have a genetic kind of map that provides us with circuitry of the brain, but during early in life, this circuitry is vulnerable, and therefore you need to see to that a child has a stimulation in vision and audition and all the other areas that a good family would give to a child. Whether we can have, people here, you have two languages, and one of the questions asked was, is it bad for the child to have both Spanish and Malaysian, or is it good? And my answer is that it's better, because the ability of the brain to recognize the different sounds is going to be is there at birth, but it's going to be maintained. If you're limited to one language, you tend to have more restricted survival of the ability to recognize various sounds, language sounds, phonemes. So I think it's a good question.
Bueno, é hora de irnos. Eu quero, diante de nada, agradecer especialmente a presencia da señora Conselleira de Educación, que reforza, sen dúbida, o glamour deste acto. E tamén quero agradecer, sobre todo, a gran asistencia que me prestou o señor decano. Todo foron facilidades para o deseño deste acto. Posiblemente haxa algún problema técnico, esas cousas que sempre pasan, pero bueno, non era a nosa intención. Espero que todo o mundo vaya satisfeito. Thank you, Dr. Wiesel, for your talk.
And I thank all of you, and I hope you all will go to Sweden and look at the beautiful nature.
Este é unha casa e nunha isla, pero moi ben, moi ben.
Thank you.